Now let's move on to look at some other components of the cell. Mitochondria are also membrane-bound organelles. However, they are not part of the endomembrane system. They're found in all types of eukaryotic cells. They're the organelle that's responsible for making all of the ATP necessary to power every function in our body. They have two membranes, the outer membrane and then an inner membrane. The inner membrane is highly convoluted and forms these things called cristae. So each of these little invaginations is a cristae. Between the two membranes is the intermembrane space. And in the very middle of both membranes is filled with a fluid called the matrix. A matrix in general means fluid with all of the components that are in it. On the surface of the inner membrane, we also see embedded proteins that carry out the functions of oxidative metabolism that we'll examine later in the semester. One thing that's really interesting about mitochondria as well as chloroplasts is that they have their own DNA. So in short, the mitochondria are the powerhouse of the cell. They produce all the ATP, that is all the fuel necessary to fuel the activities of that cell. Chloroplasts are another double membrane bound organelle. They're found in plants and some other eukaryotes. They contain chlorophyll and are the site of photosynthesis. That is, they're responsible for capturing energy from the sun and turning it into fuel. In the chloroplast, much like in the mitochondria, the inner membrane is highly convoluted, but it's organized in a little bit more complex of a fashion. It's organized into stacks of membrane-bound sacs, which we call thylakoids. Grana are stacks of these thylakoids. So here you can see three thylakoids arranged into one granum. The plural for granum is grana, so many stacks of thylakoids are grana. The thylakoids are the site of the light reaction portion of photosynthesis that we'll examine later in the semester. Now there's a theory of how these double membrane-bound organelles ended up inside the cells. Keep in mind, the original cells on Earth were prokaryotic, very simple cells, and it's proposed that some of today's eukaryotic organelles could have evolved from symbiosis between two cells that were each free-living prokaryotes. Now, symbiosis means that there's a benefit to both. So in this case, a one-celled prokaryote that perhaps was larger than the other engulfed a smaller prokaryote. And that smaller prokaryote would now be bound in two membranes. So it has its own membrane, and then by phagocytosis, the larger prokaryote pulls in the smaller prokaryote, and so it's wrapped in a second membrane. Endosymbiosis simply means endo, inside, symbiosis, living together, inside. And it's a potential explanation of how mitochondria and chloroplasts came about. Other evidence for this is that both mitochondria and chloroplasts have their own DNA. Next, we'll look at the cytoskeleton. The cytoskeleton is a network of protein fibers that are found in all eukaryotic cells. This network of protein fibers supports the shape of the cell and keeps organelles in fixed locations, as well as helps transport materials around in the cell. It's a dynamic system where components are constantly forming and disassembling and reassembling in new areas. There are three main types of filaments. There's microfilaments, microtubules, and intermediate filaments. The microfilaments are mostly actin filaments. They consist of two chains that are loosely twined together, and they allow movements like contraction or crawling or pinching, which we'll see occurs after mitosis and meiosis as cells divide. The microtubules are the largest of the cytoskeletal elements. They're a tubular structure formed of dimers, that is, pairings of alpha and beta tubulin subunits. This type of cytoskeletal component facilitates movement of a cell and materials within the cell. The microtubules can act somewhat like highways or rail lines within the cell. The final type of filament are these orange ones here, the intermediate filaments. They're partway between the size of the actin filaments and the microtubules. They're very stable, and of all the components of the cytoskeleton, these ones are generally not broken down. So you'll see microtubules and actin filaments being broken down and reconstructed in different areas of the cell. So how does movement occur within the cells? The cytoskeleton is responsible for orchestrating all the movements of the cell itself as well as the materials within the cells. The microtubules form sort of a system of highways along which molecular motors can package and transport things. Down here you can see a microtubule. This molecular motor composed of some dynein, you don't need to worry about that, but it's holding onto a vesicle. In neurons, for example, they're very long cells. One could reach all the way from the tip of your toes to almost the base of your neck. In order to get materials down to the part in the tip of your toe, if the cell is near the base of your neck, you need to transport things a long way. So these molecular motors will pick up vesicles of materials that are made up in the cell body of that neuron, and they will literally motor them down these microtubule fibers or rails to the destination where they're needed. We'll also see that the endoplasmic reticulum forms a certain amount of channels, so a protein that's destined for export might make its way through channels in the endoplasmic reticulum until it gets closest to the area that it needs to be before it becomes wrapped up in membrane in a vesicle to be transported to the Golgi apparatus. Another type of movement that we see in cells is when organelles are moved and divided and put in place for cell division. The centrosomes are regions that surround centrioles in almost all animal cells. Centrioles are found at opposite ends of the cell. The centrosome is the microtubule organizing center. This is where we see the assembly of all of the microtubules that are involved in moving things around the cells. In particular, they're involved in moving chromosomes during the process of mitosis and meiosis. We'll see how centrioles and centrosomes are related later in the semester. But basically, you can see these green centrioles. Centrioles are like the puppeteers that throw out their strings to connect to the chromosomes to move them. Around the centrioles are other materials that are involved in this arrangement. This area is called the centrosome, so I like to think of it as the centrioles operate from within an area called the centrosome. So we've addressed how things move within a cell. How about how cells themselves move? I mean, most of the cells in our body don't move, but some cells do. Essentially, all cell motion, again, is tied to the movement of actin filaments and microtubules within the cell. We might see things like cells crawling. You've probably seen video at some point of an amoeba. They're moving by pseudopod action, which is extension of the microtubules and actin filaments, pushing out a projection and then sucking in the tubules and microfilaments on the other end. Constantly building and rebuilding the cytoskeletal components can allow for these organisms to move. Some cells will crawl in this fashion with the use of pseudopodia. Other cells will use flagella or cilia. Flagella are long tails, like a sperm would have, and cilia are hairs that beat on a cell surface. Some microorganisms move around by beating their cilia. We'll also see some ciliated cells in some of We can also see ciliated cells on the surfaces of some of the cells in our body. For example, we'll see cilia on the cells in our respiratory pathways that help us remove mucus from the lungs. Now, the point here is that flagella and cilia that we see in eukaryotes and prokaryotes differ. You'll remember in prokaryotes, those tails sort of spun around, whereas in eukaryotes, they beat back and forwards. They have this 9-2 arrangement of microtubules, as compared to what we see in prokaryotes. You might remember this arrangement. The main point here is it's different than that of eukaryotes. The cells of plants, fungi, and many types of protists have cell walls that protect and support the cells. The cell walls of these eukaryotes are chemically and structurally different than the prokaryotic cell walls. Plants and protists have cellulose as a major component, and fungi have chitin. Also, plants will have primary as well as secondary cell walls that you can see exhibited down here in this figure. The cell wall surrounds the cell membrane and allows the cells to have a much stronger structure and grow against gravity without having bones like we do. Animal cells lack cell walls. However, they have an extracellular matrix, which means their membranes secrete an elaborate mixture of glycoproteins. Glyco being sugar, protein, well, you know that. So it's a combination of sugars and proteins in the space all around the outside of the membrane. You may also see that collagen is abundant. This extracellular matrix layer forms a protective layer over the cell surface. We'll also see integrins that link the extracellular matrix to the cell's cytoskeleton inside the cell. So integrins are pictured here as they go through the phospholipid bilayer, anchoring the cytoskeleton on the outside to the actin filaments of the cytoskeletal elements on the inside of the cell. This allows what happens on the outside of the cell to influence the cell behavior of the cytoskeleton on the inside of the cell. This is a table that gives you a comparison of prokaryotic animal and plant cells, and it could be useful in your study of this material. Now, all the cells in our body interact with each other. Some of them communicate through signaling, and others literally have to stick to each other. For example, the cells of our skin need to be anchored to each other in order to maintain the integrity of our skin. This is usually accomplished through surface proteins. These surface proteins will give a cell their identity as well as link cells to one another. The surface proteins that give us identity are like the MHC proteins that we see in our immune system. Those are major histocompatibility proteins. These allow us to recognize self cells from non-self cells. We'll also see cells make contact and read each other and react to each other. These cell connections are proteins or glycolipids, that is, sugars and lipids that come together. Glycolipids compose most tissue-specific cell surface markers, like our major histocompatibility proteins, whereas it's mostly proteins that link cells to one another. Let's look at some of the cell connections. There are three classes we'll examine here. First, the tight junction. The tight junction literally connects the plasma membranes of adjacent cells. We can see that image here. The tight junction forms a complete barrier all the way around the edge of the cell, preventing passage of materials between cells. So that's the most important aspect of a tight junction. It holds the cell together to the next cell so tightly the materials cannot pass between the two cells. Next, there are anchoring junctions. Anchoring junctions mechanically attach the cytoskeletons of one cell to the next cell. These are called desmosomes. You can see an image of desmosomes here. It's pretty much like a rivet. One side inside the cell that's a protein domain attaches to another protein domain on the inside of another cell. Each side literally snaps into the next. Desmosomes attach cells tightly to each other. However, they allow the passage of materials between the cells. The third type of junction are communicating junctions or gap junctions. They allow chemical and electrical signals to pass directly from one cell to another. It's literally like punching holes in a particular region, all the way from one cell through the other. So there's a channel allowed for fluid to pass through, carrying its electrolytes, and thus allowing electrical signal to pass directly from one cell to the other. So that brings us to the end of the content for this chapter. Let's have another card quiz before we wrap it up. The organelle that produces the majority of the cell's ATP is the... Which of the following is not a type of cytoskeletal filament? Which of the following does not have an endosymbiotic origin? Animal cell walls are composed of... Here are the card quiz answers: yellow, green, blue, and red. So that ties up this chapter. Please take a moment now to do your Learn Smart module as well as your diagram. The diagram for this chapter is much more straightforward and simply involves diagramming each of the cell types and their components, as well as explaining the function of each of those components.